long career. He did his PhD in Leiden University. Then he has done a string of postdocs in UC Davis, in University of Tartu in Estonia, then one in Brittany in France, and then halfway through another postdoc in India now. So he has had an international presence. He has done a lot of field work in many parts of the world, especially in Africa. So he knows a lot about butterflies, about their natural history, their ecology, and their evolutionary history as well. So I look forward to his presentation or interaction session with him about macroevolutionary history on microevolution. Yeah? It's a catchy title. All yours.
Yes. So this is working, yes? Yes. Okay. Thank you very much. So great pleasure to be able to talk to you and to interact with you here. So I chose today the topic of effects of macroevolutionary history on microevolution, because the topic of the workshop is just in between there in speciation. And I took Phytophagous insects in diverse vegetations. It's always exciting to find new information about host plant use in the wild. So this is a picture I took a long time ago. I was focused on taking the picture. Later I realized that it was laying eggs. Later I realized that it's the first host plant record for this species. So this is the kind of stuff that is natural history. It's really fun. How many of you are active in natural history? Yes, so that is always, I'm just a little bit active in it, but it's a really nice thing to do, and which can really inform the bigger science as well. So for this afternoon, I want to first talk about evolution of host plant use. I will give an introduction, then we will digest some papers together. Secondly, look at the effects of vegetation diversity on insect life history traits, some introduction and discussion, and see how much time there is to still talk about the butterflies on grasses. And that includes an experiment. And actually I wanted to start with an experiment. So Ulasa already introduced a bit about my past. So I've mostly worked there in Uganda, but I've been around. Mostly worked on butterflies, but also a bit on other organisms. So let's just start with the experiment now. So I brought some grasses and petri dishes. Yeah, you can bring this to the podium. Yeah, everything. The camera is not following me. So we'll figure out why we're doing this later on. So here there are 6 pieces of grass. So it will be very decorative actually. It's going to be a bit messy. Yeah. And in this case we have caterpillars. So a whole box of petri dishes. Yeah. So I want you to line up. Take 3 petri dishes. So
each one how many?
3.
3.
Yeah. So there's some scissors down here somewhere. Yeah. Label. A little bit improvised. So in each petri dish we do 2 leaves, 2 of different plants. So we're going to look at the choice of the caterpillars. Which leaf do they prefer? So you put 2 leaves from 2 different grasses in there, and one caterpillar, which is in this case. This grass is also included in the experiment. No, one plant species and then of another plant species. Put this together.
Anyone, right?
Yeah. So try to do different from the others. Yeah.
2 species in one dish.
Yes. Yeah. So and the caterpillars are on here. So you can gently take a caterpillar from here.
Oh,
Yeah.
that's awesome.
Yes.
Any 2 leaves in
Yeah. Try to vary that. Not everybody has the same 2 leaves.
Total there will be 2 to 6 grass species and 3 caterpillars.
Yeah. Yes.
Supposed to take a caterpillar from this one.
Yes. You take a caterpillar from here. You can also take some grass from here.
Okay.
So these are actually the same. Just took 2 because there's not much leaves on it.
One caterpillar.
Yes.
but 2 species of grass. Every dish will have 2 leaves.
Yes.
2 different species.
Yes.
Then
One caterpillar inside. So the
every,
caterpillar.
so you have a lot of caterpillars.
Well, if you run out, then it's not. But there are many small caterpillars under the leaves. Yes.
With the leaf, and this is another thing.
Yes.
I have to take one more.
So in each dish there need to be a choice of 2 different leaves, 2 different species
In each.
in each dish. Yeah. So that each caterpillar has a choice between 2 leaves.
So say the same 2 or different
Different 2 in one dish. Yeah.
from all the 6.
Yeah.
You've got a big.
Oh, she's got a big one. Yes.
Your results are going to be significant.
So one caterpillar in each dish, right?
Yes. Mhm. If you like, you can use a marking pen to write your name on it, so you can put it somewhere and still recognize later which one is yours.
Brush to remove
No, I
it.
don't have brush. Just the fingers.
I have a pencil brush.
Get
Yeah?
it, get it.
I usually use my fingers gently. Yeah, you can leave it somewhere. If you like, you can write your name on it.
I try to use this one. Try to use this.
Not supposed to do anything now. We just
We just put them there, and near the end of our session, we're going to see which leaves the caterpillar has chosen.
so there should be a caterpillar in each petri
Yes,
dish.
yes.
Are there that many caterpillars?
I hope so. Yes, there are quite many little caterpillars over here.
Here.
But if we run out, then so be it. But here's still some caterpillars. Who needs a caterpillar still?
Yeah, here there are a lot. See, 5.
I gotta use one. No, I thought
The write your name on the.
Yeah. Who else?
Take the leaf and jackpot.
Need how many? 1, 2. Is it 2? Need 3. Oh yeah. 1 and
One caterpillar in each petri dish, 2 leaves in each petri dish.
These two are different plants, right? And that's the same plant as this.
I'll get a better one for you. That one might not do so well anymore. The brush is indeed nicer.
You break the leaves, right?
Yeah. Yeah. People are still lining up for caterpillars.
You need one more, no?
Where? Here?
This one. Who need?
Yeah.
Are they gregarious?
In the beginning, they are gregarious. Yeah.
What's gregarious?
That means that they like to stay together. They look for each other's company. Just the first instar tends to be gregarious in this species.
Okay, one is the bigger one. I'm guessing 15 star. Where is
Yeah,
the
4th or
smaller one?
5th. They're mostly second instar.
Can you
Here.
help me something?
Do you need to make caterpillar?
Can you please get grasses? Okay. Any grass? 6 different grasses, like 2 in each. Who need? 3 I have.
I need one more.
You need 4.
One more. I need
They cannot take both. He has over there. See.
If not, I have to grow.
In different big.
Yeah, I have.
Yeah. Need. You can
Anybody need
keep them somewhere on the table there or anywhere you like. And if you like, you can
Anybody
put
more
your name on it
caterpillar?
so you know.
You had?
I got caterpillar. I didn't have it. I got. Everybody got caterpillars? Okay. Everybody.
Yeah, everyone has.
I think everyone had.
I'll eat this one.
Where you will keep it, Fred?
Sorry.
Where you will keep it?
Well, you can keep them just over here somewhere if you like.
All are different kind of grasses or? Yeah, different. No two are same in one.
so that they will have access, because at the end they will come and check it, right?
Yes.
So that everyone can come and see it. If you keep it here, it will be very crowded.
Yeah.
They cannot come and see.
Well, whatever works.
This one I used for
Okay.
Or we'll keep it outside.
Is it not too hot? Yeah, it's fine.
It's fine.
Yeah. But I think people like to watch their caterpillars. And
Or keep them with themselves.
yeah, yeah. Okay.
And this design will be like this. This is glasses.
Yeah, that's the lesson. Okay. Thanks everybody for helping. So we're going to look at those caterpillars later. Try not to disturb them too much so that the caterpillars can quietly go do their business. Just explore their petri dish and make a choice. So I was talking about macro and micro evolution. So let's first be clear on what it means. So on one hand, is there the pointer is in the middle? Yeah. Yeah. On the one hand, you have here rank order, and this is difference between two individuals. So in biology, the main unit is an individual organism, and the difference between one and the other organism can be very small. Like for example, me and my brother, we are very close together. Our common ancestor is just our mother, while the common ancestor can also be very far away. So the difference between two individuals can be very large, and you can measure that in the number of years or millions of years. So on this part you have a microevolution, and on the other part you have macroevolution. So microevolution is usually experimental or field studies on changes in gene frequencies and traits that might change over time that you can even study in the lab. And macroevolution is more about phylogenies, comparative studies. And then in the middle, they have this gray area from what is a species, what is not a species, and the process of speciation, which is just the border between those two fields. So I want to talk about how the macroevolution of the vegetation is affecting the microevolution of insects. First, it's good to look at the organisms. Many organisms are parasites associated with particular hosts. So when Krishnamek was here, he was talking about parasitoids of caterpillars that have such an enormous diversity. And just another example is mites, for example. Most species of bees are host to several mite species. At least 31 lineages of mites have independently colonized bees. And individual bee mite species tend to be geographically restricted. So it's not that one species has the same mites all through its range, but in different parts of its range there are also different mites. Most of these mites are quite host specific, and then you can calculate there are so many bees described, probably there are more bees than have been described, and what would be the diversity of mites on bees alone, which is already more than the number of mite species that has been described. So very many organisms are parasites. So we have ticks, we have feather lice, mites on beetles, parasitoid wasps, worms that live within your body, and also fungi. Just a few examples of parasites associated with hosts. Another example are phytophagous insects. They are parasites of plants. About half of the described species are insects that feed on plants. So let's look at plants. Plant communities have a phylogenetic structure, so they all also have a common ancestor, and so you can look at the phylogenetic diversity of vegetation. So here we have a vegetation with 3 species. Both of them have 3 species. So which vegetation has the highest phylogenetic diversity? Left or right? Sorry? The right. Why does it have the most phylogenetic diversity? Yes, it goes further back. So the diversity in terms of phylogeny is higher. Although the species diversity is the same, phylogenetic diversity is different between those 2 plant communities. So a plant species in a community has also a degree of phylogenetic isolation from the other plant species in a community. So if this dark would be your focal species, in this community it is not as much phylogenetically isolated from the other species than in this community, where the other species in the community are phylogenetically distant. The same story you can tell for an individual plant. So an individual plant has neighbors, and the phylogenetic distance with its direct neighbors can be small or large. So if this is just an individual plant with 2 neighbors, this one has a low phylogenetic isolation, and this individual plant has a high degree of phylogenetic isolation. So this is the macroevolutionary structure. So the phylogeny of plants is presented to the insects in the form of plant communities. So if you look at the forest, it's not just all trees, but for an insect, it's a macroevolutionary landscape of plants. There's not just a distance flying from here to there, but there's an evolutionary distance, millions of years that are separating 2 individual plants. So it is, depending on how specialized an insect is, which trees it can use, it's like a sort of archipelago of trees. So how is that affecting those insects that are associated with those trees? So I want to talk about how does host plant use evolve, first of all. First we need to understand that a bit more. Then how does host plant specialization affect life history traits? Third, how does vegetation diversity select for particular life history traits? And fourth, how does phylogenetic isolation of individual trees affect life history evolution? So what kinds of adaptations may Phytophagous insects have to use a particular host species? Any ideas? Anyway, if you have any questions, just interrupt. Right? So what kind of adaptations does the insect need to use a particular host?
Physiological, as in mouth parts that are strong enough or most suitable for chewing on the plant material, and also digestive adaptations to digest the plant material and make most use of it, and the other biochemical adaptations for dealing with the phytochemicals.
Yes. Any other ideas?
Also some adaptations to be able to identify that plant.
Needs to identify the plant. Who needs to identify the plant?
The caterpillars.
The caterpillars?
Or
Any others? The adults as well? Mm-hmm.
oh, sorry, yeah, butterfly.
Ranjit? Any other ideas? So this is about the finding the plant, recognizing it, and then digesting it well and growing, right?
And if
What,
there is,
what else does
if
jump
there is some
inside?
sort of anti-herbivory strategy, then you'll have to some adaptations to overcome that as well.
Yes, so that is part of this biochemistry, to be able to work with again to overcome the plant defenses. Any more ideas? Okay, let's see what I came up with so quickly. So I also thought about oviposition. So it needs to be able to recognize that particular host. Then there's larval performance. So that was part what Ravi mentioned. It needs to be able to chew it, needs to be able to digest it and have a good growth on it. But also, defense against natural enemies can play a role. So in case of chemical defense, often insects can use that chemical defense of the plant to make its own defense, to use the chemicals of the plant for its own defense. If you're a specialist, it's probably much easier than if you're a generalist. Maybe there's some other ideas about how a defense can be linked to using a particular plant species.
So if the I can use the cryptic nature or something like that to avoid the predation on the caterpillar.
Yes. So for a caterpillar, for example, it can be very important to have good background matching. So if you use many different hosts, then you will not be able to achieve good background matching. If you use only one host, then the fine-tuning of the background matching is much more likely to happen.
It could also get adapted to digesting some toxic materials, which we heard yesterday that it can get accumulated, which can be toxic to the predator.
Right, so it can sequester the chemicals that the plant is producing for its own defense. So that is also much more likely when you're specialized on a particular plant. Another part is phenology. So if the insect phenology and the plant phenology have to match, if the fresh leaves are out before the insect is out or after, then it's also not a good match between the host plant and the insect. So these are some of the aspects of host plant use, which all have to be taken into account. So how do host plant shifts happen? An insect is using a particular plant, it's doing well. Why or how is it going to change to a different host plant? Of course, it's a very difficult thing to study because it's something that you not often observe in the field, a host plant shift happening in real time. People suspect that it has to do with oviposition mistakes. Every insect now and then makes a mistake, but sometimes that mistake might be actually working out very well for it, and then it can be maintained in the population. For example, if the mistake is a genetical mistake of the recognition system of the adult, it mistakes the wrong plant as its host plant, but actually it's working out well, so that change, that mutation might spread in the population. Also, it has been suggested that sometimes in a small population, the host plant is crashed, and then the insect is there and has no other choice than to lay on something else. So it chooses the next thing available. But some mistakes are more likely than others. It's more likely to make a mistake on a different plant that provides similar stimuli, and it's also more likely to make a mistake to lay on a plant which is common rather than to lay on a rare plant. So you can expect a similar biochemistry which affects both the oviposition preference and the performance of the larva. Then similar background color, which can also affect the oviposition if oviposition is also taking into account background color, and it affects the performance or the mortality of the larvae under predation pressure, and abundance in the habitat. So if it's abundant, then it's more likely that oviposition mistakes will be made on the abundant plant. And once it shifts to there, it's more likely to persist, less likely to go extinct, because it will be able to have a larger population on that new host plant. So for us, it would be much more likely to see that before it is extinct. Another part is if they have much time together. If two species have been in the same habitat for a very long geological time, then their cumulative chance of shifting to that host plant is also larger than if a plant has just invaded a community and has only been there for a few years. Then you're less likely to see a host plant shift to that plant. So one effect of this is that closely related plant species because closely related plant species tend to be similar in biochemistry, maybe also in color, insects are more likely to shift to more closely related plant species. So the effect is then that there is a congruence between plant and phytophylogenies. So often you find that the phylogeny of the plants and the phylogeny of the insects seem to match. It's almost suggesting a co-evolution. Usually it's not really a co-evolution in the strict sense that they have been evolving together, but usually the plant lineages was there and the insect has been spreading in a phylogenetic pattern because more phylogenetically related plants are more similar, so it's more easy to shift to more phylogenetically related plant species. Yes?
You told two things. Like two possibilities can lead to host plant change. One is if the host plant in a smaller population and it is getting crossed, so then like it is more likely to shift to a related plant. But if it is a genetic change, because of that the recognition system is getting changed, then it may not, like it is not necessarily to be a similar plant, it could be any plant, isn't it?
It could be any plant, but if you would expect that it's still looking for something that is similar. Although it's changed, it's unlikely that it changed to something completely different, more likely to something slightly different. So it's still more probable to shift to something that is more similar. Yes?
In plants, are grasses more similar in between species? Do they have similar phytochemical signatures, or in general they are less toxic?
Well, in general, grasses are probably less toxic. They're more relying on mechanical defense and also on regrowth. But still, also in plants, there are some chemicals. Take, for example, lemongrass, which has a very strong chemical in it. Plus, grasses often have endophytic fungi in them that produce alkaloids. And the alkaloids can also affect the herbivores. So together with the fungi, they can be quite toxic. Okay. So the last bit I would propose to talk about grasses. So another result of this is that more closely related plant species harbor more similar insect communities. So here you have a community of closely related species, and the insect communities are also similar. Here's a community of species that are more distantly related, and the insect communities are also more different. So it has been shown, for example, here in a forest in New Guinea. Here's the plant phylogenetic distance for any pair of two plant species, and the insect community similarity. So in the triangles are congeneric species pairs. So the species that are very closely related, they tend to have a high similarity. Then these squares are species not in the same genus, but in the same family. So the similarity in the insect community is already less. And when the two individual plants compared are from two different plant families, then the similarity in the insect community is also less. So that is one part, the phylogenetic signal, but also the species have to occur in the same habitat. It's unlikely to shift to a host which actually occurs in a different habitat. So here you have the records of tree species in geological time. So how common has this tree been, and the number of insect species associated with it. So the higher the abundance in geological time of the species, the more insects have colonized that plant. So here are some new arrivals that have just recently arrived, and these are species that have been abundant for a long time, and they also have more insect species associated with them. So as I just said, also their arrival time, not just abundance, but arrival time also plays a role in geological time. As was just shown here, there are some special symbols. These pluses are of introduced plants, which tend to have fewer insects associated with them. But also if you look at invasive plant species, we can actually see how invasive plants are accumulating more and more species over the decades. So I think we have explained that already. So how do you study such a kind of food webs? One way is to select a certain number of plant species and make an inventory of all the insects that are feeding on it. So again, here in the forest in New Guinea, here are the plant species, 38 species of plants. And here are different types of insects that feed on them. So here I have mobile chewers, it's mostly grasshoppers and stuff. So the grasshoppers, they can feed on really many plants. It's not really a parasite, as what I was talking about before, but all the others are more or less parasites. For example, here we have caterpillars, the exposed chewers. So each caterpillar is connected to the plants it can use. So the more insects are associated with a certain plant, the bigger the bar is, and the more plants a certain caterpillar species can use, the broader the band is here as well. So you see there's most caterpillars are specialized, or they use a few species, and most trees have several species of caterpillars feeding on it. If you go to miners, then you get much more specialization. And for mesophyll suckers like aphids and cicadas, it is typically very specialized. And why do you think that miners are more specialized than caterpillars? So leaf miners live inside the leaf rather than on the leaf. Yes, have idea, Alex?
Something to do with the physiology.
With?
Something to do with the physiology.
Yes, so when you live inside the leaf, you deal much closer to the physiology of the leaf, and you have to be more in tune with that plant. So that requires a higher degree of specialization. And on the other side, we have semi-concealed caterpillars. So these are like leaf rollers that roll the leaves, so they are a bit protected from predators. And they are less specialized than the exposed chewers. Why do you think that could be? Background matching. Who said that? You said that? Yes. I think that is a good idea, I think, because if you're rolling yourself in a leaf, your own color doesn't matter. So that strategy can work on a wide variety of hosts. So the common picture is that in a given community of caterpillars, for example, there are many specialists that use just 1, 2, 3, or 4 host plants, but there's always a tail of a few generalists that can use very many species of host plants. Okay, so what time is it now? 2:45. Okay. So the next part is about some activities. So we have, I will give a demonstration of paper reading and presenting. Then we'll assign some papers and then we can have some tea. So what I would like you to do is to make groups, read a paper together and present it using 20 minutes of your time. Yeah? So you cannot actually read the paper, but it's about like quick reading of a paper. Who is used to that strategy of reading a paper without actually reading it? You? Nobody else? Okay, so it can be a good exercise. And it is indirectly endorsed by Ulasa, so don't be worried. So I'm going to cheat a little bit because I'm going to do it with a paper that I've already seen. But because it's such a cool paper, I just wanted to do it. I didn't actually read it recently. So we just have the... Oh, you can't see my screen? Maybe I have to... I was assuming that my screen would be visible, but maybe I have to go out of this mode here. Yeah? Say, how do you do that? You can just see my screen and not the PowerPoint. Yeah. And that's not the way, I guess. Oh. You know? Sorry?
This is the display also.
Oh yes, there we are. Thank you so much. So here's the paper. It's called Spatio-Temporal Variation in Leaf Miner Population Structure and Adaptation to Individual Oak Trees. Spatio-temporal variation in leaf miner. So I'm especially interested in the second part, adaptation to individual oak trees. So we're going to edit, take a snapshot of that. Does everybody know the snapshot tool? And yeah. Oh, now I did this again. Okay, there I am. I just want to make a new PowerPoint presentation. Okay, that's fine. New. So there's my title of my PowerPoint presentation. Oh, that is too much. Sorry about that. I guess you're all much better at this. Yep. Okay. And then we go to the PDF. So then we have an abstract. Maybe you have time to read the abstract. Maybe not. And then we go into the figures. So we see here a picture of a lake with trees around it, and then there is some drawing, and the drawing shows some different trees. So there is a young extinction young control, mature, and dead. So dead should be this kind of dead trees. Young extinction, it means that they killed all the insects on that tree. And then there's a control where they did not do that, and then there are some mature trees that they didn't use because they're too big to kill all the insects. So if you want to present this paper, you'll have to take the picture. And put it in the slideshow. Okay, so that is the methods. I'm cheating a little bit. You will need a bit more time to realize that. Then there's math. Math you always skip. Math and statistics, you never look in it if you don't have time, unless you're interested in the math itself, if you want to do math as well. But if you're interested in the ideas, and you don't usually have time to follow that. So this is going to be important. This young extinction, young control, and mature over the years. So we have 1990 up to 1997. Leaves with leaf mines. So in this case, they killed, after 1990, they killed all the leaf mines. And then they came back. So the population came back. And here in 1997, somehow there were not many leaf mines. So in this time period, the population rebounded from this killing. That's all they want to say here. And this is something about gene flow. I was not so much interested in it. Here you have, so I'll just skip that for now. I just want to know about this adaptations. Okay. Some genetics. Table, let's skip that for now. Okay, here is a graph. So in the graph, I can make it a bit bigger perhaps. So you have the 4th, 10th, and 40th generation. Now these light things are something about allozyme structure, which is before the DNA work was easier to do. And people used allozymes to look at genetic variation. And here's fitness. So what fitness in this case means is that they transplanted one mine to another tree, another individual tree, and looked how it was performing there. So they found that in the beginning it didn't really matter much. If you would transplant a mine from one tree, individual tree, to the other, usually fitness was lower because the transplantation was not very nice for it. But after 40 generations, the leaf miner always wanted to stay on the same individual tree. If you would put it on a different individual tree of the same species, it would perform much poorer than if you would transplant it within the tree. Slide. Yes. Okay. So when then what we need to do is to what are the questions. And then the last slide, we have again the questions, and we give the answers. Yeah. So I'm not going to do this for you right now with this paper, but this is how you conclude. So we have 4 slides that we created after reading the paper in, was it 5 minutes? I was struggling a little bit. I'll give you 20 minutes, so I expect you to do much better than me. Okay? So now okay, who's going to come? Yeah. So now we can make some groups. So how many people do we have? 1, 2, 3, 4, 5, 6, 7, 8, 10, 17. Am I right? Maybe I will make 4 groups. So that would be four groups of 4 people. So we'll just focus on the plant-insect co-evolution for now. So let's just do it like this: 4 people, 4 people, 4 people, 4 people, and maybe 5 if necessary. And then you can come over here, and if you have a thumb drive or something, then I will give you the paper to read. Then you can read and make together a small presentation, and after the tea break we're going to see what these papers are about. Yeah? Okay. Yes?
You can email me.
I can email you? Okay, so I'm online here. So everybody's going to see my emails. Sorry? You got already? No, this one's. So I have here, you can also find it, yeah. There's with Nillen, Slove and Jans 2014, Hammond, Fordyce 2015, and Niemann and al. I can copy for you.